everybody. My name is Kevin Bender. I am an associate professor at the University of California, San Francisco. And my lab has been studying the loss of function side of SCN2A for the last couple of years. And we've actually gone through and looked at a number of different levels, working with our colleagues at UCSF, including Stefan Sanders and Nadava Hituv's lab. We've been working going from the gene itself through the channels, the NAV1.2 channels that are encoded by the SCN2A gene, and then trying to understand what's going on in brain cells and circuits that are affected when you have a loss of function condition, and I'll explain what loss of function means in a little bit. And then also looking at animal models with the hope of translating everything that we learn at these basic biology levels into treatments and therapeutics for your children. Obviously, that's the most important thing. So when Leah asked me to do this, she asked me to just give a little bit of a background on what we've been doing over the past couple of years and give a little bit of a history of where we've been and where we're going. We started this work probably about 2016, 2017. What we have been moving towards is really just understanding what's going on at all of these levels. So I'm going to give a relatively data light but interpretation heavy presentation today, and that's because a lot of what I'm going to talk about today has already been published in peer-reviewed journals. But really, what I want to help you understand is what do we know now, just within the past 5 years, in terms of how SCN2A disorders are affecting the brain, and where we really can go, and what the future looks like. This really opens up an opportunity for us within autism spectrum disorder to really understand what is autism spectrum disorder in some respects. By understanding SCN2A, we can actually understand a lot about what's going on with autism spectrum disorder. But before we get there, I want to talk about the particular effects and the particular variants that are associated with SCN2A that give rise to these different phenotypes that we're seeing within the family group. Either severe epilepsy that starts very early in life, or intellectual disability, developmental delay that we start to notice a little bit later in life, usually before the first year, that can or cannot be associated with late onset seizure. And so to do that, we did a lot of work to correlate and to compile all of the data that we know about what are called gain of function missense mutations that are associated with various forms of seizure in these children. And you actually find that there are two forms. There's a case where there's something called benign infantile seizure, which is very relatively mild and remarkably resolves with age. And then there's a more severe form, which is called epileptic encephalopathy, which we find very prominently in the family group, that unfortunately does not resolve with age. These are due to what are called missense mutations, which change the particular amino acids, the protein makeup of this channel, and changes the properties of this channel in a very subtle but very important way. And so what you're seeing here is, if you can imagine, this is a sodium channel folded around. If you unfold it, you will find that the channel looks like this, where these little dots right here are the 4 amino acids that form the sodium selectivity filter, and that allows sodium to go in, and only sodium. And then around it are a bunch of transmembrane domains that sense voltage and just basically package the whole thing around that voltage around that selectivity filter. One of the things that's really important is this 4th transmembrane highlighted in cyan here, cyan. I don't know. I grew up in New Jersey. I can't pronounce anything correctly. This is a region that is the voltage sensor for the channel. So as a membrane of a neuron depolarizes and you start to fire action potentials, this is what twists and allows a sodium channel to open. And you can see that a lot of these variants that are associated with epileptic encephalopathy and benign infantile seizure in red and yellow, or in green and yellow here, are very close to that voltage sensor. And they change the ability or the way in which the channel responds to voltage. And typically what they do is they make the channel open too quickly. They make it open too early, but there's a subtle difference. There are those that make it open a little bit early, and then there are those that make it open way too early. And so this difference in the sensitivity of these variants that give rise to these two different disorders really corresponds to how much they change the channel's properties. And there's this little line here that is a dashed line associated with SCN8A 1.6 excitability. I want to talk about this real quick. So with benign infantile seizure, these are remarkable because they resolve with life. And the question is, why do they resolve with life? Well, one thing that's really interesting in the brain is that in some parts of the brain, NAV 1.2 has a particular role early in life, and then its role switches later in life, and NAV 1.6, SCN8A, takes over that early role. And it just so happens that the differences in the inherent properties of those channels make it so that small perturbations in NAV 1.2 or SCN2A are completely accounted for by SCN8A. And so if you haven't passed this threshold in terms of your ability to change with in terms of your variant with SCN2A, and it's not any more severe than the quote unquote wild type or normal condition with SCN8A, you tend to get benign infantile seizure. The far more severe ones that are even more excitable than this NAV 1.6 channels, those are the ones that give rise to epileptic encephalopathy. Okay, so these are all the cases where we have what's called a quote gain of function, where the channel is more active than it should be. There's another side of the coin, and that is loss of function, where the channel is not as active as it should be. This is what tends to give rise to autism spectrum disorder, intellectual disability, and then those seizures that occur later in life. Of course, there are exceptions to this rule. They are all over the place. But I would say that this general first order approximation model accounts for the large majority of the variants that we see within children. So these ASD-associated variants are different than those that give rise to seizure. In particular, you'll notice that those that are missense, they actually pop up in different places. Instead of being in the voltage sensor, they pop up near this pore, near these 4 amino acids that are the selectivity pore. What they do is instead of making the channel more active, they actually block the channel's pore completely, and the channel can't function at all. And so that's what happens with a lot of these cases. There are other cases that are protein truncated cases. What those are things that happen basically anywhere within this channel that are a bit of instruction for the intracellular machinery of the cell to tell the cell that, hey, this protein wasn't made the way it normally should be made. Instead of putting it into the membrane, let's actually just break it down and try again. But what happens is, since the instructions always say, here's a break, what happens is that the channel is never made. And so with the 2 alleles that you have from the parents, only one of those alleles actually leads to a functional channel. And this is a really common form of loss of function where you have half as much channel as you should. And so that's something called haploinsufficiency. Okay? So in our lab, we've been really interested in trying to understand what goes on when you have a loss of function and how, when you go from a channel, how does that affect the brain? And this is what we've been principally interested in our lab. But before we get there, I wanted to just talk about the incidence at which these things occur. So from estimates of when these variants are occurring and how common they are, one thing that my colleague Stephen Sanders tells me all the time is that it's so much easier to break a protein than it is to alter it. It turns out that the protein truncations occur much more commonly. And they actually occur on an order of about 7.5 per 100,000 births. Let's not talk about benign infantile seizure because they're resolved, and luckily those kids go on to lead a normal life. But let's contrast it with what we see with epileptic encephalopathy, where we see about 1.4 cases per 100,000 births. Right now, already you're going to see a mismatch. So this population should outweigh this population by a factor of 5 to 1 or so. But that's not reflected in the parent group. And I think the reason for that is that what we're picking out right now within the parent group is probably those children that have loss of function, but they tend to develop seizure later in life. Because those are the children that are going to go in and have some sort of genetic screen done to identify an SCN2A disorder. That ends up being a minority of this population. And what we're really missing right now are those children that develop intellectual disability or are diagnosed with autism, but never develop a seizure. And those children are less likely to get genetic screens, but they're certainly out there. And it turns out that if you combine these two populations here, it's about, 8.9 per 100,000 births. If you broadcast that into what we're seeing in terms of the number of births in the US every year, honestly, the family group should have 340 new additions per year, which is remarkable. But if you think about the entire world, it's 11,000 per year. So this is a very large, it's a dramatic patient population that I'm excited to see you guys are pushing for. And there are far more of you than you realize. That's really what this is trying to say. Okay, so given that we have such a large patient population, what can we do? The first thing is to really understand the basic biology of what's going on with loss of function. Where do you want to study that? And what animals do you want to use to study that? Well, we use mice. Mice have these little brains, and they're studded with SCN2A. Each of these little dots is a cell that expresses SCN2A. So up here is a very large olfactory region in a mouse because they care about sniffing. And then there's this area called the neocortex, which is involved in higher order cognition, much lower order than a human, but a mouse gets by. There's this region called the hippocampus where SCN2A has already been studied, where what we find is that, this is Tom McHugh's lab at Riken, they find that deficits in SCN2A here interfere with short-term memory, interfere with navigation, which is an important feature of the hippocampus. And then there's this other thing back here. This is the cerebellum. It lies very low on the back of your head. This is involved in motor function. This is involved in actually some forms of social interaction that are very, very important for allowing a child to know that they're doing things correctly and to update information within the brain. It's a learning center. Honestly, all of these regions are a learning center. And what I really want to tell you about is that at the end of the day, no matter where SCN2A disorders manifest, and particularly loss of function, we see evidence of deficits in learning at the level of individual neurons. Okay, so one thing I want to mention is here in the cerebellum, this is actually a really important region of the brain, and we're just starting to look at this in the lab with your help, with the family group's help. So Chenyu Wang is a graduate student in my lab. Actually, you can see in the background, Perry Spratt is just defending his thesis. He's done a lot of work on SCN2A as well. This is our socially distanced thesis defense that we've had to do. Chenyu's been building this crazy system here that looks like it's coming out of Clockwork Orange or something. But what this is a little mouse will sit here, and this table right here will rotate back and forth, and that engages an eye reflex that is cerebellar dependent. And then this screen full of these bars actually will move back and forth as well. And so the animal will have a case where its screen and its body, there will be a mismatch, because under normal conditions, you want to do this, and you can't stop yourself from doing this. This is a reflex of eye movement. We have a hypothesis that this will be altered in SCN2A haploinsufficiency or heterozygous mice. And the real advantage of this is, I think, what our hope is if we can start to study that in mice, this is something that we actually could use in the clinic, because you can track these same eye movements in a human, and since they're innate, since they're reflexive, you can actually track those same eye movements, even in a child that's nonverbal, where it's hard to give them instructions. And so we might be able to use this later as a diagnostic for when we get some therapies going. Are those therapies working? Can we have something that's quantitative that we can use to say, yes, this therapy is working? Because unfortunately, sometimes for these sort of validation things, we can't just rely on the parents saying, yes, it's working. We have to have something quantitative, which honestly, I don't believe in, because parents can't be the best of judges. Okay, so getting back to this idea, of what's going on in the brain. We've actually spent most of our time, we're just moving into cerebellum now, but we've spent most of our time in neocortex, which is a region in the front of your brain. So for example, prefrontal cortex, it's involved in higher order learning and processing. And they have these cells that look like this. They're called pyramidal cells. They're studded with these little dots right here. And each of these dots is a synaptic input. So this is a source of communication between cells. And the long and the short of it is that these dots under normal conditions are what are called plastic. They're modifiable. They can change with experience, and you can test that in the lab. And one of the things that we have found is that under normal conditions, we can change the strength of those synapses just very easily. So this is the baseline strength in black of one of these synapses. And then with a particular protocol, we can change that strength. We can enhance it for a long time, 50% stronger or 50% weaker, that sort of thing. But when you have a loss of function in SCN2A, you've lost that ability to change your synaptic strength. And so this change in synaptic strength actually underlies learning at the cellular level in your brain. And so what we're seeing here is that the system actually has a harder time learning. That's what I think is happening. And that also manifests in a couple different ways. And what looks interesting is it's not really that the system has trouble learning. It's actually that it just hasn't matured yet. Under normal conditions, you have these little spines that are these sites of input. Under normal conditions, in a healthy brain, you'll see that these spines end up looking like mushrooms, real fat mushrooms that have large heads and small necks. And you can see an example of that here. These spines start out as being these long tendrils, long filopodial things that are reaching out, searching for partners, searching for connections to make, to form, and then to strengthen. What we find in the SCN2A case is that we have a far more of an excess of these immature looking spines, and also these physiological measurements, these squiggly lines down here, they tell us that not only does the anatomy look wrong, but the physiology looks wrong, or looks immature, in that the synapses just aren't as strong as they should be. But the real important thing is they're there. So the synapses are there. And so all of them are here, and basically they're waiting for instructions. And so this is the thing right here. The number of spines per unit length of a dendrite is the exact same between a wild type mouse and an SCN2A heterozygote mouse. I would imagine that the same is true in a human. And so one way of thinking about this is that the hardware is there. The connection, the ability to form these connections is present, but the software needs a bit of an update. And the software update that we need is a restoration of SCN2A. Now the question is, when do you want to do that restoration? Okay, so this gets into this idea of, okay, let's say magically we have a therapy tomorrow, with a COVID vaccine tomorrow. What is the window of opportunity within a child's life to get some sort of restoration? Where if I have a 10-year-old child with an SCN2A disorder, if I give them a therapy, is it going to help? Is it going to help at all, or is it going to be a perfect restoration? So under those conditions, we really weren't able to answer these questions in the mice that we had, because these mice either grew up with 2 normal copies or 1 altered copy of SCN2A. What we really needed to do was to take some sort of genetic control over whether or not the animal had normal copies or 1 truncated copy. And to do that, we developed some new mice. So the first thing that we did was, you can do these fancy genetic tricks in mice. You can actually have a mouse that starts as like very normally, having 2 normal copies, and then whenever we want, we can remove one of those copies via some genetic tricks. What was interesting in those mice is that when we did that genetic trick at what's called postnatal day 30, 30 days after a mouse is born, it's actually already an adolescent. It's a 12-year-old running around hating their parents' mouse. And it actually ends up having physiology that looks identical to these mice that have grown up their entire life lacking one of their 2 alleles. And so we interpret that as meaning that SCN2A is required for brain function throughout your life. And if you lose it somehow later in life, you will end up starting to have deficits in your ability to learn within the brain. And that's what we're seeing with these two mice. Now, that's interesting because it says that anytime in life, if we can change things around, we might be able to see, we see regression. Well, what about the opposite? Can we see restoration? And so we made a different mouse, and that is a mouse that starts as a heterozygote. It starts as a child affected with loss of function, and then we're able to restore it anytime we want. And so far in the lab, honestly, we haven't done an exhaustive study yet. These mice look a lot like their wild-type brethren. And so that suggests that if SCN2A can be restored, brain function should improve to some degree, even if it's done later in life. Obviously, I think at the end of the day, the earlier we identify and the earlier we diagnose and the earlier we treat SCN2A disorders, the better. But this opens up the possibility that we might be able to see even partial benefits later in life. Okay, and really that opens up the idea of, can you start to think about therapies? Okay, so we've been working on one in the lab, and that's with Nadav Hitub's lab, with Serena Tamora and Navneet Matharu. And that is a technique to basically, in this case, where you have not as much SCN2A gene as you should because you've lost one of these alleles, can you tell this allele to work twice as hard to match this condition? And this is something where we're using a technique called Crispr activation. And so here you've got your SCN2A gene. It's being regulated by these upstream genetic components. They're either called promoters or, unfortunately misspelled, they're enhancers. And what you can do is you can take this CRISPR technology, which a lot of you know about in terms of CRISPR editing, making particular mutations within or making particular changes to the DNA. We're not using it to change the DNA. We're actually using it just as this chaperone, just as this shuttle, to bring something else to this region, and that's called an effector. And we can put that effector can either be a repressor, which would reduce output of that gene, or it could be an activator, and that's called CRISPR-A, or CRISPR activation, where it basically says, all right, you tell this gene to work twice as hard. And this is the way we're going about it for gene therapies for SCN2A. The reason for this is that Nadav's lab is a world expert in this sort of technique, and they've actually shown that it already can work for other cases of haploinsufficiency. So for example, if you have this gene called SIM1, if you don't have as much SIM1 as you should, it actually leads to morbid obesity in mice and in humans. But a simple treatment of CRISPR-A one time in this mouse, in a particular part of their brain, reduced their weight and converted them back into what looks like a wild-type weighted mouse. And so CRISPR-A has a number of features, a number of advantages, I think, over traditional CRISPR gene therapy. There actually is no DNA editing, so you don't have to worry about off-target effects. It could work for all the protein truncating variants that we're seeing in SCN2A, which actually ends up being the majority of the variants. And it takes advantage of this endogenous regulatory machinery. And what that means is if a cell expresses SCN2A, SCN2A will be upregulated. If the cell doesn't express SCN2A, nothing will happen. And so you let the cell's own instruction manual tell CRISPR where to work. And in theory, and this is the big hurdle right now, a single administration could be effective throughout life. The real trick within the field is getting that single administration into a child. Okay, so all of this is to basically show that we're able to get it working at the level of a mouse. We can increase expression in a haploinsufficient case or a heterozygote case. We actually can increase expression in a wild-type case. Not that you would want to. And if we look at features of the cellular physiology under those cases, we're seeing a rescue, somewhat partial rescue, back to normal ranges. And then we're looking at other squiggly lines, and we're seeing a rescue from if you compare the wild type to the heterozygote, you see that this heterozygote actually returns back to normal cases here, and this is synaptic function. And so that's pretty darn exciting for us, suggesting that we can get restoration. And this injection, importantly, was done around P30. So this is a relatively late in life injection of CRISPR-A. Now, the problem with this sort of approach is you worry about this situation. You worry about going from here not just back to normal conditions, but actually going too far and going all the way past this dotted dashed line into epileptic encephalopathy. That's the thing that we're really concerned about. And so one of the things that we've been testing is when you upregulate, what happens when you overexpress these channels? Does the excitability of the neuron change in a way that's actually detrimental? What we're finding so far is that that appears to not be the case. And the way we've done that is take advantage of those wild type animals, those normal animals, which already have normal levels, and overexpress SCN2A far higher. But when we do that, even though we see that the protein is at a higher level, the cell's excitability is not at a higher level. So there's some sort of ceiling effect that we're seeing here. And this might actually broadcast to a lot of different gene therapies where you can overexpress, but there are other things that are happening in the cell that help those sodium channels get into the membrane that are still doing their job. So basically, under normal conditions, if you reduce SCN2A function, you can see a reduction in protein levels at the membrane. If you increase the number of channels, it actually doesn't change the excitability of the cell. What you need instead is a gain of function variant that actually changes the biophysics of that channel to produce a gain of function case. And so this actually is quite enticing because it suggests that if we could get this therapy working, it should be efficacious and should have a good safety factor. Okay, so now I want to talk a little bit, if I have a few more minutes, about the outlook of where we are and where things could go in the future. So if you're thinking about a bunch of different potential therapies for SCN2A, you're going to think about drugs, you got to think about pharmacology that you're going to be giving children, and you also have to think about potentials for gene therapy. Leah asked me to talk about this. I am by no means an expert in all of this, but this is just my general view of where things stand right now and potentially where things are going in the future. So in terms of pharmacology, I think what we're seeing in the field is that it's far easier to block a channel than to make a channel work better. And so that means that gain of function variants are going to be prime targets for pharmacology that is specific for NAV1.2. And it looks like things like that are moving along very, very well. Potentially, I think progressing to phase 2 or phase 3 trials probably within a year or two. And so I'm very excited about that for gain of function. I think that that's a great way of looking at treating gain of function disorders in SCN2A. For loss of function, since we can't directly target the channel, you have to start to think about it, whether there are indirect approaches for loss of function. That's a little trickier, but it's something to consider. So you can block other channels that are active at particular times that could counteract the loss of NAV1.2. That's a little more outside the box, and so that ends up being a bit of a boutique industry right now. So I don't know if there's going to be much push there. But with all of these, you have to have some considerations. Pharmacology has a bunch of advantages in that you can adjust the dosage whenever you want. If it's not working, you just take it off. It's really non-invasive, but you do have to worry about off-target effects. And in particular, the things that people are concerned about in the field are sodium channels are important for your brain, but also for your heart, for your muscles. You don't want to have effects where you block sodium channels in the brain and then also block sodium channels in your heart, introduce arrhythmias in a child. That would be bad. And so the field is working very, very hard to try and mitigate those sort of off-target effects. On the gene therapy side, there's actually a number of different things. Unfortunately, there's some bad news. And the first thing is, if you think about traditional gene therapy, if you wanted to just upregulate SCN2A by just saying, let's give the child more SCN2A, unfortunately, we can't do that in our current world because the SCN2A gene is just too darn big for traditional gene therapy. So we have to think about these other approaches. Many of you have heard of what Stoke Therapeutics is doing with SCN1A and Dravet, where they're targeting what's called a poison exon. That's a great approach. Unfortunately, SCN2A just doesn't have one, and so that can't be translated over to SCN2A, unfortunately. And then this idea of making precise cuts with CRISPR to do precise gene editing, I think in the future that will improve, but the current state of the art is, unfortunately, that CRISPR gene editing is still too imprecise. And even an imprecision at the level of 0.00001%, honestly, is too much risk for gene editing, because that's going to live within your body throughout life, and it's those imprecise cuts are just going to arise further and further and further. And so that kind of technology needs to improve. But the good news is that there are many companies, many labs pursuing oligonucleotide approaches for both gain and loss of function. Some of these are moving along very well. Chris Ahern, I think, is talking in this RX Hope symposium. He's got a great technique here for looking at protein truncation. CRISPR-A, I think, avoids many of these issues that we're seeing with gene editing, because we're not doing gene editing. We're just doing gene modification in terms of taking the endogenous program and just changing the software, not changing the hardware, just changing the software. But there are considerations with that, and so there's invasiveness. So with oligonucleotide therapy, you have to give a child an intrathecal injection every 2 to 3 months. There's specificity for disrupted alleles. So for example, if you have a gain of function case where you have one normal allele and one altered allele, the current state of art with oligonucleotide blocking therapy is to dampen both of those, when in fact what you really want to do is just dampen the altered allele. So developing better techniques to actually have some specificity there would be really nice. And last is penetrance within the brain. So all of these things have to get into the brain somehow. Right now, oligonucleotides get into neocortex really well, but other parts of the brain, they don't get in very well. The same is true with CRISPR-A, which is done via adeno-associated virus. Getting it across the blood-brain barrier and actually getting enough of a dosage but before you have liver damage is a trick. All of these things are being thought about, and they're not just being thought about for SCN2A. There is a major industry for basically all gene therapies. We have to solve this for all gene therapies in the world. And so the hope is that SCN2A will be able to piggyback on all of that research and move forward. Okay, so with that, I want to stop and thank absolutely everybody in the lab, and all of the funding. In particular, you guys are now supporting us directly in the lab. Very grateful of that. This is also very strongly supported by the Safari Foundation, the Broad Institute, Autism Science Foundation, all sorts of people helping us along this way, and a lot of collaborators that have been helping us hopefully make a bit of progress here. Okay, so thank you very much.
I'm Jenny Burke. I'm a board member of the family's SCN2A Foundation, and I really appreciate Kevin being with us today to talk about his work on SCN2A. So we just have a couple questions. First of all, will your technique that you've been working on in your lab for so long work for other variants besides the protein truncating loss of function variants?
Yeah, so CRISPR-A, how hard is it to redesign CRISPR-A to work for missense variants? It's probably is a hard question. It actually is a hard thing to answer. So right now, no. The way CRISPR-A is designed is it targets both promoters that are upstream of the SCN2A allele, and those promoters are similar but not identical between the different parents. But we have a generalizable thing right now that just upregulates both. But since one leads to protein truncation, nothing extra happens there. If we're going to get to a case where we have, let's say, a gain of function variant in SCN2A, what you would want to do is block one channel and upregulate the other. Does that make sense? And that would require a redesign of CRISPR-A. It could work in the future, but I think protein truncation is going to be the first step. And then the question is, can we redesign it, and probably in a patient-specific way, to get more specificity. And so we have to know that it's going to work for loss of function before you do the even riskier project of going after a gain of function case.
All right. So can you talk a little bit about the variability of the of SCN2A patients and how treatments may be different? in different cases?
Yeah, so I guess we talked about that a little bit with the last couple of things. If you have a gain of function case, I think the things you should be looking for right now are pharmacology coming to bear. I think the way in which pharmacology might be improved in the next couple of years is actually quite promising. And the same would be true for antisense oligo approaches, where you can blunt that activity. If it were, it's hard to know which one. Basically, we have a situation where every approach should be considered right now, and we don't know which is going to be best. And it might be that that one works better for a particular child compared to another child, and so everything is on the table at the moment.
How could an SCN2A family get involved what's the best thing we can do as families to support your work or get involved in?
Well, I'd say you guys are already supporting our work, and we're very, very grateful for that. We are pursuing things with direct support from the Family Foundation that that I'm hoping will lead to a better understanding of the basic biology in the brain, but also some development of what are called quantitative biomarkers to assay whether there's a particular disorder and whether or not that disorder is rescued with therapeutics. And so I'd say, keep doing what you're doing. It's great.
That's great. And what would you say is the time frame for getting to clinical trials?
Well, I would say for our own work with Crispr-A, we're still a ways off. We still have to do work at the basic biology level, the pre-clinical trial level, that validates this approach, that it works in a mouse model, that we would probably want to be using other animal models as well. And so that I would say is a long ways off. Even that's the stock, oh, it's 5 years away. That's not even true, I would say.
No way.
But things like pharmacology, things like antisense oligonucleotides, those have already gone through a lot of safety metrics in the world for other disorders, and so I think those are far closer. You have to remember that CRISPR-A simply hasn't been done in any human for any disorder, and so SCN2A is not going to be the first of those, but we're hoping to basically be in the pipeline, ready to go once other people have proven that it's safe and effective therapy.
Great. Would you say we should encourage people who have autism spectrum disorder, but no seizures yet, to get genetic testing?
I would, and the easiest way to do that is to enroll in Simon's Searchlight. They are doing the testing for you for free. Sometimes the returns are very slow, and they'll only return information if they actually have a hit. But all it requires is a little bit of spit from everybody.
Should we encourage people that we know who have autism spectrum disorder to get genetic testing?
I definitely would. And the easiest way to do that is a program called Simon's Searchlight through the Simons Foundation. And the advantage there is you don't have to worry about your insurance, trying to fight with your insurance, and it's free. What happens there is that everybody in the family gives a little bit of spit, and then that gets sent off to Searchlight, who does the genetic testing for you. And if you end up finding a hit, they will let you know about it. If there is no hit, which actually is probably more common, you may never hear back from them, but certainly it's worth it, especially for hocking a loogie in a bucket for a few seconds. I mean, what's the hardest thing? That's not the hardest thing to do.
Right, that's great. Well, thank you so much. We really appreciate your time to keep us informed about what's going on.
Yeah, thank you so much for having me, and everybody stay safe.
Thanks.